Namaste students, first of Vidya, I, JP and Dr. Vaishnavi, Assistant Professor, Jyoti VIDYAPEETH, University, Jaipur. In today's session, we are going to discuss about the gene mapping. Gene mapping is the sequential allocations of the glucans for relative position on a chromosome. That means genetic map are the species-specific and comprised of the genome marking or the genes and genetic distance between the each marker. And here the chromosome mapping, when we say, it is a technique used for the autosomal DNA testing, which allows the testing of to determine which segment of the DNA comes from which ancestor. That means in order to map the DNA segment on the specific chromosome, it is necessary to test a number of those family relatives. So what are the basis of genetic mapping? That means the genetic mapping looks at how genetic information is shuffled between the chromosome or between the different regions of the same chromosomes during meiosis, or a process called the recombination or crossing over. So why is crossing over is for chromosomal map is important here? In genetic mapping, which we call it as linkage mapping, okay? So can also be firm evidences for which that is transmitted from parents to the children and is linked to one or more genes, okay? So here the mapping also provides a clue about the which the chromosome contains the gene or precisely where the gene lies on the chromosome, okay? So here, how many genes are in a chromosomes? Normally, when identifying the gene on each chromosome is an active area of the genetic research. That means because of the research used between different approaches is predicted and the number of genes on which the chromosomes or the estimated number of the genes vary. That means the chromosome 1 likes to contain 2000 to 2100 genes that provide the instruction for making the protein, right? So here, what is the first chromosome? That means the chromosome 1 is designation of the largest human chromosome. Humans have two chromosomes and one as the down autosome, or which is the non-sex chromosomes, and one span about 149 million nucleotide base pair, which are basic units of the information of the DNA. And first 100 years ago in 1913 has laid the foundation of the modern biology. Who gave the basis of the genes or possibles in 1919. So here mapping means that the patch or the point on the object of the match of the both other sides. Okay. So the gene mapping describes the method used to identify the locus of the gene and the distance between the genes. Okay. So which is essential for the genome mapping is placed in a collective or a collection of a molecular marker onto their respective positions on the genome. Okay. So here the molecular marker come in all forms, that is the genes can be viewed as one special type of the genetic marker in the construction of the genome. Map or the map that's based on the other marker. Genome marking or the genome mapping is based on the or used of the genetic techniques to construct the map showing the position of the gene and other sequences of the genome. So the genetic technique includes the cross-breeding experiments or the case of the human or the experimentation of the family space instruments or the physical mapping or use of the molecular biological techniques to examine the DNA molecules directly in order to construct the mapping showing the position of the sequences, features, or includes the genes. Okay. So the genetic mapping, that is the first step of the building of the genetic maps, are the development of the genetic marker. So here the mapping for population is closer to the marker of the chromosomes. Okay. So here you can see the it is likely that it can pass on to the next generations also. So there is a correlation or co-segregation pattern was taking place. Okay. So the marker can be used to construct their order and the genotypes of the each genetic materials are recorded for both parents and each individual in the following generation. Okay. So the quality of the genetic material is largely dependent on the two factors. That means the number of the genetic marker used in a map or the size of the mapping. Here the first step is the building of the genetic map after the development of the genetic marker. Okay. So here in gene mapping, any sequence or the features can be faithfully distinguished from the two parents can be used as the genetic marker. So the genes are represented by the traits. That means they can be distinguished between the two parents and their linkage with the other genetic markers are calculated. Some ways as if they are common markers and actual genes, the loci are then bracketed in a region between the two nearest neighboring markers. The entire process is then repeated by looking at more markers which targets the region to map the gene. So here there is a high resolution until a specific positive locus. Okay? So here the physical mapping, that means restriction of the mapping which locates the relative positions of the DNA molecules on the recognition of sequence for restriction endonuclease. Okay. That means the fluorescent in situ hybridization, that is FISH, the fluorescent in situ hybridization, which is a marker located on the map by hybriding a crop containing a marker to intact the chromosomes. Okay, so sequence tagging site SDS mapping, in which the position of the short sequence are mapped by the PCR and the hybridization analysis of the genome fragment. Okay. So the physical mapping or the physical mapping can be generated by the restriction map of the specific piece of the clone or the genome DNA, for instance, in line or VSC vector along the chromosome. These maps are extremely useful for the purpose of map-based clone gene cloning. Okay. So when we see the genetic versus the physical distance, the map distance between on the recombination frequency are not direct measurement of the physical distance along the chromosomes. So here the recombination hotspots or the overestimate physical length or the low rate of the hypochromatin or the centromere administration texture physical length. So what are the use of the gene mapping here to identify the gene or responsible for the disease, heritable diseases, cancer, or identifying gene responsible for the trait. That means plant versus animal or the milk productions. So many human genomic projects are being taken place where the human gene projects as an international scientific research projects with the goal of determining the sequence of the chemical base pairs or having a different identical genes on both physical and the strand points. Human gene project originally have the nucleotides here. Okay. Limitation by genetic techniques is barely sufficient for directing the sequencing phase. This is for the two reasons, that is the resolution of the genetic material depend on the number of the crossing over and have been sensor possible. And so there the gene or the several tests they appear as the same position on the map. So here we can see the distance, physical distance between the chromosomes. So So here you can see when it is a genetic and the physical versus the physical distance. The physical map, when we say it is the here the heterochromatin, and the distance of the man using the genetic maps. Okay, and here you can see these are the genetic maps for the physical distance and the map position. Actually, that means a very little recombination take place in heterochromatin part. Okay? That means a small distance of the genetic map. When we see the genetic map, it is the small distance, right? This is the here it is the small distance. Okay, right? This is the small distance. So this part is a small distance. Okay, 54.5 to 57.5. But when we say it is the physical distance, this is the physical distance. So the genetic map corresponds to the last distance of the chromosomes. Thank you. This session is powered by 2.0 JVWU Men's University, Jaipur. If you have inquiries, we'll discuss in the problem solving session.